And the degree of saturation, i.e. how many double bonds that are present, will affect that particular fat's melting point, whether it's solid or liquid at room temperature. The more unsaturated fatty acids, the more liquid the fat will be at room temperature. The more saturated the fatty acids, the more solid the fat will be at room temperature. So solid fats tend to be more saturated and thus less desirable for health. Liquid fats tend to be much more unsaturated and thus more desirable or preferable for health. Okay. So in figure 5.6, it's showing us fatty acid composition of common food fats. So most fats are a mixture of saturated, monounsaturated and polyunsaturated fatty acids. So take a look at the composition of fatty acids found in coconut oil. So that's going to be the top one here on this figure. So coconut oil, marketed as a health food and healthy fat choice within the food industry. But note that it's largely composed of saturated fatty acids, which we know are less desirable in the context of cardiovascular health. Conversely, look at flaxseed oil, or otherwise known as linseed oil. It's a much healthier blend of predominantly unsaturated fats, and actually is a great vegan source of essential fatty acids, omega-3 and 6. So see here, the orange type colour is representing the proportion of omega-3 fatty acids, and then in the blue, omega-6, green monos with a very low proportion derived from saturated fatty acids. So arguably a much better type of fat choice within the diet. So now we're going to move on to talk about the other two categories of lipids, namely phospholipids and sterols. So phospholipids are structurally similar to triglycerides in that they also contain that glycerol backbone, but they have 2 and not 3 fatty acids attached. Instead of the third fatty acid, a phosphorus molecule is attached to the glycerol backbone, and actually the presence of that phosphorus molecule renders the whole structure to be both soluble in water, thanks to the phosphorus, but also soluble in fat, thanks to the remaining structure. So we can sometimes use the word so soluble in water can sometimes be described as something being hydrophilic, so it likes water. And being insoluble in water can be described as hydrophobic, dislikes or afraid of water. So just remember hydrophilic and hydrophobic as we move on. Because of its solubility both in water and fat, phospholipids naturally aid fats to be dispersed in water. So they're acting therefore as emulsifiers. Okay. So in figure 5.7, the oil and water within the flask, without the help from emulsifiers, fat and water separate into layers as shown here. Think of in food manufacturing, emulsifying agents are added to blend water and fat-soluble ingredients together. A really, really common one is something called lecithin. That's spelt Lecithin. And this is a type of phospholipid used to blend vinegar with oil in things like salad dressings or mayonnaise. So that's one example of an emulsifier. Phospholipids also play a crucial role acting as cell membranes, owing to their dual solubility in water and fat. So they bind together, forming a strong double layer which acts as a membrane to cells, with a hydrophilic phosphoric or phosphate head and a hydrophobic glyceride tail. So what you end up with essentially is a double layer of phospholipids, where the 2 fatty acids from each phospholipid are facing one another, connected to the glycerol backbone, and then the phosphate molecule, or the phosphorus head, are the furthest away from one another on the outer most side of each side of the cell membrane. If you're not quite sure about that, quickly Google phospholipid cell membrane and you should get the idea. But we don't need to go into any more detail than that. Okay. Moving on to sterols, the third type of lipid. Sterols are large, complex molecules consisting of interconnected rings of carbon atoms with side chains of carbon, hydrogen and oxygen attached. So an example of a sterol is cholesterol, So cholesterol serves as a raw material for making emulsifiers in bile, for example. Cholesterol is also important in the structure of cell membranes of every cell, and so it's essential for the body to function properly. We need it for optimal cellular processing and structure. Cholesterol can be made in the body, so it's therefore not considered an essential nutrient. Other sterols examples include vitamin D. So vitamin D is not actually a true vitamin, as you'll learn in chapter 7. It's actually made from cholesterol. Also, certain steroid hormones such as estrogen and testosterone are also examples of sterols. Okay. So the next learning outcome was just recapping on the digestion and absorption of fat. So let's recap. So in the stomach, after being chewed and swallowed, food travels to the stomach, as we know, where droplets of fat separate from the watery components and tend to float as a layer on the top within the stomach. Even the stomach's powerful churning can't really completely disperse the fat, so very little fat digestion takes place in the stomach by gastric lipases. So once it enters the small intestine, bile made in the liver is, as we know, stored in the gallbladder, is released into a duct that leads to the small intestine, where it's needed for fat digestion. So it's released on demand, i.e. in response to the fat-containing meal. And bile emulsifies and suspends the fat droplets within the watery fluids, the chyme or chyme, until the fat-digesting enzymes provided within the pancreatic juices can split them open into smaller fat molecules for absorption. So that's what we know already about bile. Okay, so triglycerides are split up into a combination of free fatty acids, phospholipids, and monoglycerides. So monoglyceride, think that's a glycerol backbone with one fatty acid attached, monoglyceride. And as we know, the bile may be reabsorbed and reused in the body, i.e. recycled, or it may flow back into the intestinal contents and be lost within the faeces when bound to dietary fibre. Recall that concept covered in the previous chapter. So regarding fat absorption, at the intestinal lining, fat fragments in the form of free fatty acids, glycerol and monoglycerides, are absorbed by intestinal villi. Glycerol and short-chain fatty acids may enter directly into the bloodstream, where they may then travel to the liver. So these molecules are small enough or soluble enough in water to do so. So this is where our knowledge expands upon what we learnt within chapter 4. So let me just repeat that for you. Glycerol and short-chain fatty acids may enter directly into the bloodstream, where they then may travel to the liver. Okay, so although I told you before, mostly all fat-soluble nutrients travel initially in the lymph, we're expanding upon that knowledge now. So these molecules, this is why that's possible. They're either small enough or they have sufficient hydrophilic properties. They are soluble enough in water to travel within the blood. The cells of the intestinal lining convert large lipid fragments, such as monoglycerides and long-chain fatty acids, back into triglycerides. So they convert them back into a triglyceride structure, and then they combine them with protein to form a new structure, and this is a new concept, which are called chylomicrons. So that's spelled CHYLOMICRONS. A chylomicron is a type of lipoprotein. And chylomicrons may then travel within the lymph vessels, and that's how larger, more insoluble fat-based nutrient molecules are repackaged, enter the lymph, are transported and then delivered into the blood. Chylomicrons are therefore the transport form of larger fat molecules, as I've just explained. They initially travel in the lymph and are eventually delivered back into the blood from the lymph and circulated within the blood, as chylomicrons themselves are soluble in water. Okay, so this is illustrated in figure 5.8. Everything the initial stages of what I've just said. So Bile and detergents are both emulsifiers and work the same way, which is why detergents are effective in removing grease, or think about Dawn or dish soap when you're trying to scrub an oily pan. It's the same concept. So molecule by molecule, the grease is dissolved within that dirty sheet pan, for example, and then suspended in the water when it can be rinsed away down the drain. So if you think of the action of bile, this is what this figure is illustrating. So from the left, fat and watery digested juices tend to separate, as we saw in that, I guess, in a different way with the fat droplets suspended in water. So without emulsification, enzymes are in the water and can't get at the fat. They can't take action and start breaking down triglycerides into smaller fat fragments. So in the presence of bile, delivered from the liver, stored in the gallbladder via a duct into the small intestine, bile compounds have an affinity, so they really like both fat and water, so bile can mix the fat into the water. Okay. After emulsification, more fat essentially is exposed and made available to the enzymes so that fat digestion can continue. All right. This slide, I want you to pause yourself. I've gone through all of the text, so this should be recapping for you. This figure is available in the textbook on page 154, or you can enlarge the image from the PowerPoint if you pause from your downloaded version of the slides. Have a recap, read this information yourself to go over the theory again. Okay, so moving on. So the transport of fats depends on the size and solubility of the fat fragment, I guess. So glycerol is water soluble. Okay, shorter chain free fatty acids, and they're called free because they're not bound to anything else. They can pass both through the intestinal lining and enter the bloodstream, i.e. they do not travel via the lymph, whereas longer chain fatty acids, phospholipids and monoglycerides, they're either too large or too insoluble in water, so they must be repackaged into chylomicrons. Again, that's spelt there on that slide for your reference. And chylomicrons are a form of lipoprotein, i.e. protein bound to lipid. Once remodelled, chylomicrons first must travel in the lymph, and eventually they drain back into the bloodstream for distribution. So that's just recapping on everything we've just said. Okay. So storing and using the body's fat. So as we know, our subcutaneous adipose tissue, our fat stores, act as a storage facility for energy in the form of fat. And these stores are made from dietary fat supplied in excess of our immediate needs, as well as from carbohydrate. Remember that when there are adequate total calories from carbohydrate in the diet, and the supply is greater than demand, i.e. our glycogen stores are full, the liver will convert carbohydrate to fat for storage. And therefore, in a time of need, so a call of energy, such as starvation or severe illness, stored triglycerides from within those adipocytes, those fat cells, are broken apart, releasing free fatty acids into the bloodstream for immediate supply of energy. Cells can either break down these fatty acids into smaller fat fragments, which within the cell may be combined with a carbohydrate fragment, and that then enables that structure to be burned for energy in the usual cellular cycles and processes. Without adequate carbohydrate supply, remember from the previous module, whether the carbohydrate is stored or being delivered via the diet, so in an inadequate carbohydrate supply, the products of incomplete fat breakdown in the form of ketones, remember ketones, they then start to accumulate in the tissues and blood, and may also be excreted in the urine.